Hey, my name is Kayla Steen, and I'm on almost PGY4 at the University of Alabama at Birmingham in radiation oncology, and together we have been going over radiobiology for the Radiologist. This has largely been based on the 7th edition of the text from Hall and Giacca, but recently obtained the 8th edition, have been making some changes accordingly. A little about me, I'm currently a PGY3 at the University of Alabama at Birmingham, and I earned my PhD in molecular biology with a focus in cancer epigenetics from UAB in 2019 before deciding to go to medical school. I used to teach and tutor advanced molecular genetics while in graduate school, so I really do love this stuff. What the ABR says we need to know. So the box section is the second half of the Hall and Giacca textbook that's meant just for radiation oncology residents. I've already made videos on the first half of the textbook that's for both radiology and radiation oncology residents. And we are nearing the end of our topics that we need to know for the board exam. Here's the table of contents from section 2. And today we're going to be covering the biology and exploitation of tumor hypoxia. So jumping right in, hypoxia inducible factors. We've kind of talked about these in brief in some of the previous lectures. These are going to be transcription factors that facilitate both oxygen delivery and adaptation to oxygen deprivation by regulating the expression of genes that are involved in many cellular processes, including glucose uptake and metabolism, angiogenesis, erythropoiesis, cell proliferation, and apoptosis. So the structure of hypoxia inducible factors it's a heterodimer composed of 2 subunits. Okay, there's the alpha subunit, which is the oxygen sensing subunit, and then the beta subunit, which is the ARNT subunit. That's the aryl hydrocarbon receptor nuclear translocator. There are 3 families of HIFs. There's HIF1, HIF2, and HIF3. So a little bit more about the individual families. HIF1 binds an 18 nucleotide fragment of the erythropoietin enhancer required for the hypoxic activation of erythropoietin and Hep3B cells. So the generation of more red blood cells in response to hypoxia. So the analysis of the alpha subunit, and again, the alpha subunit is the oxygen sensing subunit, reveals 4 distinct domains, including an oxygen-dependent degradation domain, or the ODD, that regulates HIF1 alpha degradation by the ubiquitin proteasome pathway. HIF1 is ubiquitously expressed and induces the expression of most of the hypoxia-inducible genes. Next, we have the HIF2 family, and this shares about 50% sequence homology with HIF1. Expression of HIF2 alpha is restricted to endothelial cells, glial cells, type 2 pneumocytes, cardiomyocytes, kidney fibroblasts, interstitial cells of the pancreas and duodenum, and hepatocytes. And then HIF3, not really well understood in vivo, but responds to hypoxia response elements in vitro. Now let's talk about the oxygen-dependent regulation of HIFs. So in the presence of oxygen, HIF1 alpha has hydroxylated on 2 highly conserved proline residues, both found within the ODD. Again, that is the oxygen-dependent degradation domain. The enzyme collagen-4 prolyl hydroxylase uses molecular oxygen and 2-oxoglutarate as substrates with iron and ascorbate as cofactors. Hydroxylation is required for binding to the VHL tumor suppressor protein. This leads to destruction of HIF alpha, so the alpha subunit of any of the HIFs, by the proteasome. Okay, so let's think here. The presence of oxygen, okay, oxygen acts as a substrate for the enzyme to work and deactivate HIF1 alpha. Okay, deactivation is performed by the proteasome. So it's going to be destroyed by the proteasome. And this is done by VHL. So von Hippel-Lindau tumor suppressor protein recognizes the hydroxylation, and this leads to destruction by the proteasome. But in hypoxic conditions, this 4-prolyl hydroxylase doesn't have that molecular oxygen as a necessary substrate. Therefore, HIF1 alpha can't be recognized by VHL. This is going to result in protein stabilization. Then HIF1 alpha dimerizes with the beta subunit and subsequently binds to a core sequence and the enhancer elements of target genes to initiate transcription. This is figure 26.1 from the textbook, and this is a visualization of what I just had written on the previous slide. So in the presence of oxygen, okay, we do not have hydroxylation of HIF, okay, and so then HIF can be translocated to the nucleus and dimerize with the beta subunit, resulting in transcription of hypoxia genes. But in the presence of oxygen, we see hydroxylation of HIF from this prolyl hydroxylase. The hydroxylated HIF is recognized by VHL, and then HIF is degraded by the proteasome. We can also see HIF being transcribed and translated via this other PI3 kinase pathway, okay, that is oxygen independent. So additional regulation of HIFs. We can have recruitment of co-activators. There is this third hydroxylation event of HIF alpha subunits on the C-terminal transactivation domain on the asparagine residue 803 by asparaginyl hydroxylase, also known as the factor inhibiting HIF, okay, the FIH. This takes place under normoxic conditions, preventing interaction with P300, which prevents gene transcription. We can obviously have some cancer mutations that activate HIF, okay? And the most obvious of these is going to be a mutation in the von Hippel-Lindau protein, resulting in von Hippel-Lindau inactivation. This leads to HIF stabilization and increased target expression irrespective of oxygen concentration. So if you don't have VHL or you have a non-functional VHL, then you're not going to have the proteasome degradation of HIF. This is going to lead to the development of highly vascularized neoplasms like hemangioblastomas of the retina and central nervous system, RCCs, endocrine and exocrine pancreatic tumors, and pheochromocytomas. We also have inactivation of PTEN, which if you saw in the previous slide from that previous figure from the textbook, figure 26.1, PTEN blocks the PI3 kinase pathway. And so if you remember, the PI3 kinase pathway leads to transcription and translation of HIF. So if you have inactivation of PTEN, you're going to have increased HIF activity through the mTOR or PI3 kinase signaling pathway. So how does HIF help tumors? We can have tumor angiogenesis, right? If you're in hypoxic conditions, you're going to need more blood vessels to promote delivery of oxygenated blood. So HIF induces VEGF, which has potent pro-angiogenic properties, if you remember from one of my previous lectures. HIF also helps with tumor metabolism. So we have this Warburg effect where tumor cells shift to the glycolytic pathway and away from the oxidative pathway. HIF-1 regulates the expression of glucose transporters, glycolytic enzymes, lactate production, and pyruvate metabolism. Again, all of that moving towards the glycolytic pathway and away from the oxidative pathway. HIF can also assist in tumor metastasis. HIF activation results in transcriptional regulation of E-cadherin, lysyl oxidase, and CXCR4, and all of these are going to govern cell adhesion, extracellular matrix formation, and cell migration. So this is figure 26.2 from the textbook, and this is kind of a summary figure based on my previous slide. So HIF-1 alpha, okay, it affects lipid metabolism, it affects the oxidative pathway, right, the electron transport chain. It's going to affect the oxidative pathway of cellular metabolism. And it's also going to affect the transport of pyruvate into the mitochondria for the TCA cycle. Okay, so HIF-1 alpha is going to result in and the inhibition of pyruvate dehydrogenase, which is required for transition into the TCA cycle. So therefore, it's going to promote glycolysis. It's going to affect glucose transport into the cell, and then the lactate dehydrogenase pathway. So now let's talk about HIF and radiation. We see this reoxygenation dependent increase in HIF-1 activity. Release of reactive oxygen species leading to increased HIF stabilization and translation of target. So if you remember from my way earlier lectures about ionizing radiation, we have the indirect action where we see the formation of reactive oxygen species, which causes DNA damage. Well, those reactive oxygen species leads to increased HIF stabilization and then further translation of targets. So it promotes tumor vasculature radioresistance through release of VEGF. This dominates because HIF-1 deficient tumors are more radiosensitive. It also can induce radiosensitivity through the induction of apoptosis. So if you think about the way that radiation induces cell death, that's going to be mitosis-related cell death versus the hypoxia cell death is going to be more so through apoptosis. Next, we have this unfolded protein response. So just remember, in cell biology, we just have a bunch of redundant pathways such that if one doesn't work, then most of the time there's going to be a backup. So in this case, prolonged periods of hypoxia can activate non-HIF signaling pathways like the unfolded protein response. So the UPR is a cellular stress response that is induced by the accumulation of unfolded proteins in the endoplasmic reticulum in order to deal with the problems associated with the accumulation of misfolded proteins. So the unfolded protein response can cause transient inhibition of protein translation to prevent further misfolded proteins. So if the proteins aren't folding correctly, well, we don't want any more of those, so we're going to stop protein translation. The UPR can also induce chaperone proteins that function in the endoplasmic reticulum to assist in folding, trafficking, and the secretion of proteins. So if we have these proteins that are misfolded, then we're going to have some chaperone proteins that help fold. Okay, so we're going to prevent folding or prevent protein translation so that way we don't accumulate any more misfolded proteins. And the misfolded proteins that we do have, we're going to have chaperone proteins that function to assist in appropriate folding. We have this protein called IRE1, and this is an ER stress sensor that's going to become activated through homodimerization and trans-autophosphorylation. It has endoribonuclease activity that splices the pre-mRNA of the X-box binding protein, or XBP1 transcription factor, that results in its translation. So XBP1 has many target genes involved in ER protein maturation. So IRE1 becomes activated through homodimerization and trans-autophosphorylation, and it's going to result in the translation of XBP1 that helps in ER protein maturation. So a little bit more about this unfolded protein response. We have the protein kinase-like endoplasmic reticulum kinase, or PERK, and this is a second ER stress sensor. So if you look at our picture over here, we have PERK, and it's located within the membrane of the endoplasmic reticulum. So it's going to prevent further translation of proteins, and activation occurs rapidly under severe hypoxia. You can see that we have oligomerization and autophosphorylation that leads to its activation, and it's going to inhibit translation by directly phosphorylating serine 51 of eIF2 alpha, and this is going to be a key initiator of the mRNA translation machinery. So it's going to inhibit the activation of eIF2 alpha. So this is going to be important for surviving hypoxic conditions. So how do we radiosensitize hypoxic cells? One of the more obvious answers is hyperbaric oxygen, right? So if you introduce patients to just very high oxygen concentrations, you can make these hypoxic cells now normoxic. But this is very cumbersome and it comes with poor compliance because these patients have to go here daily for very, very long periods of time. And we've seen attempts using carbogen, and so carbogen is oxygen plus 5% carbon dioxide. The whole goal here is to improve oxygen supply to tumors, all right? So we can do that with blood transfusions, right? If you physically have more hemoglobin, you physically can transport more oxygen to the tumors. And so you can give blood transfusions prior to radiation. And you can also use artificial blood substances such as perfluorocarbons. And then you can also have something called a hypoxic cell radiosensitizers. These are going to be compounds that are not rapidly metabolized by the cells through which they diffuse, and then they can penetrate further than oxygen and reach the hypoxic cells in the tumor. So Adams in the 1960s developed some criteria for these compounds. So what are going to be some properties of compounds that make them good hypoxic cell radiosensitizers? So they're going to selectively sensitize hypoxic cells at a concentration that would result in acceptable toxicity to normal tissues. So we don't want to have to give so much of this radiosensitizer that it's killing normal tissues. It also needs to be chemically stable and not subject to rapid metabolic breakdown. And it needs to be highly soluble in water or lipids and must be capable of diffusing a considerable distance through a non-vascularized cell mass to reach hypoxic cells. Okay, it needs to be able to diffuse 200 microns. So from previous experiments, this rind of normoxic cells or cells that are functioning under relatively normal oxygen tension, it's going to be about 200 microns. Okay, so this necrotic or hypoxic portion of the tumor is about 200 microns from the nearest capillary. So it's got to be able to diffuse a considerable distance from that capillary into the hypoxic portion of the tumor. And it should be effective at relatively low daily doses of a few gray used in conventional fractionated radiation therapy. Here are some examples of radiosensitizers. So interestingly, we found that one of the first radiosensitizers was actually flagyl or metronidazole. And so metronidazole is a nitroimidazole, okay? And so we had further development of other nitroimidazoles from metronidazole or flagyl. So the first of these is mesonidazole, and this is a 2-nitroimidazole. So just giving you a little bit of biochemistry here. So the side chain determines position 1. So this is a nitroimidazole. Okay, and the side chain here is just as basic as it could be. It's a hydrogen. Okay, so the side chain determines position 1. And if the nitro group is in the second position, then the drug is a 2-nitroimidazole. If the nitro group's in the fifth position, the drug is a 5-nitroimidazole. And just in general, 2-nitroimidazoles are better radiosensitizers. So mesonidazole is a 2-nitroimidazole, and there was a Giaccia study that demonstrated an advantage in male patients with high hemoglobin levels and larynx cancer. So if you look at the overall study, it was a negative study, but if you do this subgroup analysis, it showed that in male patients with high hemoglobin levels in larynx cancer, there was a benefit. But there was dose-limiting toxicity that was peripheral neuropathy, and this actually progressed CNS toxicity if the drug was not stopped. So think like myelopathy type stuff. Then we had the development of etranidazole. And so this medicine is a 2-nitroimidazole, and it is less toxic, but it was shown that there was no benefit. So the doses could be increased by a factor of 3 compared to mesonidazole, but there was no benefit. And then we have the development of namorazole, okay? And this was less active, so you could, and it's much less toxic, so that large doses could be given. And the Giacca trial showed that there was a significant improvement with local regional recurrence and survival, but we still don't see it given very often. So let's talk about other ways that we can increase the oxygen content of a hypoxic tumor. So nicotinamide and carbogen breathing. So you can overcome chronic hypoxia with carbogen breathing because we see more delivery of oxygen to the tumor. And then we also have nicotinamide, which is a vitamin B3 analog, and this prevents transient fluctuations in tumor blood flow that lead to acute hypoxia. Remember, acute hypoxia is where we have just transient aberrations in these blood vessels, right? Because tumor blood vessels just are not as good, they're not as stable. And nicotinamide can actually prevent these transient fluctuations in tumor blood flow. There was the Archon trial that I talked about in a previous lecture. And so it utilized accelerated radiotherapy, carbogen, and nicotinamide. And so this showed it was accelerated to overcome proliferation of the cells in between fractions. It was hyperfractionated to spare late responding normal tissues. So, the dose was split up into more fractions. All right. And then carbogen breathing was used to overcome chronic hypoxia. And then nicotinamide was used to overcome acute hypoxia, excuse me. Now let's talk about hypoxic cytotoxins. So we were just talking about radiosensitizers. So radiosensitizers in hypoxic conditions are going to be different. All right, hypoxic cytotoxins are going to kill the hypoxic cells, not radiosensitize them. Examples of these are going to be quinone antibiotics like mitomycin C, nitroaromatic compounds. We have benzotriazine dinitrooxides. They're like tirapazamine, and we're going to talk more about that. We have dinitrobenzamide modified nitrogen mustards, and we have 2-nitroimidazole attached to a dibromo isophosphoramide. So let's talk about tirapazamine. So this is going to be a hypoxic cytotoxic compound, okay? So the time sequence here is giving the drug before radiation. And so looking at skin reactions, all right, that there was not an increase in radiosensitivity with normal tissues. That's very important, okay? It selectively was killing the tumor cells. Side effects of tirapazamine are going to be nausea and severe muscle cramping. And there was a trial, TROG 0202, and that compared tirapazamine plus cisplatin plus radiation to cisplatin and radiation in stage 3 or 4 squames of the head and neck. And it showed that there was no significant difference in failure-free survival, time to local regional recurrence, or quality of life. But if you're looking here, kind of in the more in vitro studies, you can see that there was, a really big difference here in the surviving fraction of aerobic cells to hypoxic cells. So this shows that the tirapazamine was selectively killing the hypoxic cells. So now let's talk about targeting tumor metabolism in order to kill hypoxic cells. So we know from previous slides and previous lectures about the Warburg effect. This is the shift to the glycolytic pathway from oxidative phosphorylation in cancer cells, regardless of oxygen status. It's just been shown that tumor cells exhibit altered metabolism that's highly glycolytic at the expense of oxidative phosphorylation. And so looking at the HIF1 pyruvate dehydrogenase kinase axis, or the PDK1 axis, is one of the major regulators here. It leads to phosphorylation and inactivation of pyruvate dehydrogenase. This is the first committed step for pyruvate entry into the mitochondria. So if increasing mitochondrial activity in moderately hypoxic cells results in increased hypoxia through metabolizing oxygen, well then decreasing mitochondrial activity in hypoxic cells should result in increased oxygen levels, right? You're preventing the pathway that utilizes oxygen. So if you look back at one of the figures from one of the previous slides, figure 26.2, it just shows that, if you target the electron transport chain, there's the terminal electron acceptor oxygen, and this takes place through a series of redox reactions. So, inhibiting the electron transport chain is generally, the best way to increase radiosensitivity, right? Because it's going to result in decreased oxygen consumption. So just thinking in general, the mitochondria is going to be the key target in altering oxygen consumption within a tumor cell. But there's this drug called DCA, or dichloroacetate, and this is an inhibitor of PDKs, and it's been used to treat patients with inborn errors of metabolism. DCA increases mitochondrial metabolism in these patients by inhibiting the activity of PDKs. So DCA has actually been used in preclinical tumor models and mouse models to increase mitochondrial function, and it reprograms tumor metabolism to be more oxidative like normal tissue. So it's going to stop the shunting of pyruvate into the glycolytic pathway and lactic acid formation, and it's going to shunt it more towards oxidative phosphorylation. So at the mitochondrial level, at the molecular level, DCA can inhibit PDH phosphorylation. Okay, it can reduce glucose uptake, and it can increase mitochondrial function and inhibit tumor growth. And again, this is in model tumors, I think within mice, the mouse model. So DCA can enhance the efficacy of hypoxic cytotoxins such as tirapazamine. Okay, without causing depression of hematologic parameters. So there's clinical trials investigating the efficacy of DCA to alter tumor metabolism and then increase tumor hypoxia. And you can kind of follow this using a PET scan, right? Because an FDG PET scan utilizes radio-labeled glucose. So you can actually look at those metabolic changes within the tumor. And that is all I have for you guys on the biology and exploitation of tumor hypoxia.